not quite there yet. One more minute, please.
No worries. I can see that it shows its webinar is now streaming live on Facebook.
I can now confirm we're live on Facebook. We're good.
Good.
All right. Thank you, Bill. Good evening, everybody. We're excited to have you join our October virtual Lyme impact session. We've been really wanting to do this particular topic for quite a while. And there's really no better individual that can kind of lead us through the quagmire of Lyme testing in America than Dr. Burrascano. My name is Eric Huck. I'm the president of PA Lyme Resource Network. Joining us this evening is Bill Moore. He's board member, IT director, and Bill runs the Pittsburgh Lyme support region in Pennsylvania. And as many folks know, PA Lyme has now grown to 20 support group regions, all in Pennsylvania, all volunteers. And we seem to be doing a lot of right things lately in terms of networking, working with ILADs and legislation. What I'd like to do is, before turning it over to Dr. Burrascano, I would like to give a little bit of an update on where we are with Lyme legislation in Pennsylvania. Not only Lyme legislation, but also statistics. The statistics that we have received are for 2022. And every year from the CDC, it's usually about a year after reporting is when data starts filtering down. But Pennsylvania has led the nation in the number of Lyme disease cases 11 of the past 13 years. In addition to that, in 2022, there was 8,413 reported cases of Lyme disease, and this is based on insurance claims. The CDC, as most folks now know, they estimate that to be 10 times higher. We believe the multiple probably is higher than that, but we're willing to use the CDC's 10x multiplier. That's going to equate to 84,000 cases of Lyme disease in Pennsylvania in 2022. Really, the staggering statistic is 25% approximately are children ages 5 to 14. This translates to about 50 children a day, and estimates are anywhere from about 10 to 25% become long-term chronic sufferers. And that equates to 4,200 children a year contracting Lyme, having chronic long-term symptoms, and having challenges and illness for as long as it's going to take for them to treat and eradicate that. So when we focus on children in Pennsylvania, and that really is kind of a benchmark with PA Lyme, a lot of the work we do centers around a focal group of how do we prevent children from getting infected? What can we do for education? How can we get into the schools? How can we interface with school nurses? I'm happy to report that Senate Bill 232, the Lyme disease education of parents and establishing protocols for tick bites in schools, it has actually moved very quickly. For those of you that are on our email distribution, or you're a part of our Facebook community, you got a notice within the last 3 days that it was time to call your House representative. And the sponsored bill is by Senator Michelle Brooks. And there at the top, this requires school officials to notify parents about the removal of a tick. If a child at school has a tick, there's a protocol of how to remove it, either through a nurse or a teacher designated at a school. This creates a mechanism for the school or the parent, if the parent chooses, to preserve the tick and send it to the Tick Research Lab of Pennsylvania for testing. And then upon getting the results of the test, the school will notify the parents. This is super important because early diagnosis, early recognition that there is a tick that carries a disease, the early testing of a child, the early treatment of a patient goes a long way to reducing the time that people are effective and the ramifications of their illness going forward. Very happy to say that this passed the Senate Health and Human Services Committee unanimously. It went to the full Senate vote where it passed. It was kicked over to the House Education Committee where it passed 19-6 about 10 days ago. And then it just passed the full House vote, 179-23. Once it clears the House, goes through a formal step, it'll go to the governor's desk for signature. We are working with Senator Brooks' office to determine should we have a call to action of the Lyme community that's in our network to be sending letters or emails or phone calls to the governor's office. We haven't gotten a clear direction. We will probably know within the next day or two. So be on the lookout. It's entirely possible you're going to get exposed to another either email request call to action or on Facebook or Instagram, Twitter, what have you. So what we have heard is the grassroots aspect of PA Lyme pushing our legislators for sensible Lyme legislation goes a long way. So thank you to the grassroots that really contributed to the success of this getting out there. So with that, I would like to introduce Dr. Joseph Burrascano. He's a physician who was an early innovator in the field of tick-borne diseases, having begun his clinical research in the mid-1980s. He is well known for his pioneering treatment guidelines for Lyme and related tick-borne illnesses. It became a classic series that has been freely circulated around the globe since 1989. He's a founding board member of ILADS, very important, International Lyme and Associated Diseases Foundation. He also served as a director of the ILADS Educational Foundation. He continues to be active in educating practitioners and the public on clinical diagnosis and treatment of people with Lyme and related. He has authored many articles, many book chapters, a lot of papers on tick-borne diseases. He's advised the CDC and the National Institute of Health. He has also testified before the U.S. Senate and various governors' councils. Dr. Burrascano is a graduate of the New York School of Medicine, a specialty in internal medicine. He currently works full-time as a project analyst and manager for a private biotech company. He's helping to bring advanced technologies and treatments out of the lab and into the general use for the public. He's dedicated to the education of practitioners, patients, and other stakeholders in this Lyme community that we find ourselves. So Dr. Burrascano is pleased to join us today to discuss the clinical and laboratory diagnosis of Lyme and related tick-borne diseases. One last thing I would share is prior to COVID, most of our regions had monthly meetings, local support group meetings, and that worked very well until COVID hit. We had to make a decision to spin to be more remote and more virtual. So we created the series that you're participating in tonight, the Virtual Lyme Impact Series. And our very first featured speaker was Dr. Joseph Burrascano in the late spring of 2020. So here we are 4 years later, welcoming him back. And with that, Dr. Burrascano, thank you for joining, and may I turn it over to you.
Well, thanks, Eric. Thank you very much. It's my honor to really be here looking at the hard work you've all done and what you've accomplished. So I'm honored to be here. So thank you for inviting me. I'm going to share my screen now, get the presentation going. Little by little, I'm learning the technology. Here we go. All right. And I'm going to minimize this picture of ourselves. Good. All right. Of course, I started with the last slide. Here we go. Technology, you see. There we go. All right. So I've been asked tonight to discuss like the real world of tick-borne disease testing, what the tests are out there, which are the good ones, not the good ones, and when to use which test. When you have a situation, an early illness, a late illness, and so forth, which test you use and why. So we'll get started right away. The first two slides, this one and the next, actually set the stage for this whole entire presentation. And you have to understand the history of Lyme to understand why we've had so much trouble with testing. Turns out that when Lyme was first discovered to be an infection by a spirochete, Borrelia burgdorferi, that was accomplished because Willy Burgdorfer came to an island off of Long Island, New York, not too far from where I live, and he collected ticks and found this spirochete in the ticks. This one spirochete that he found became the prototype lab strain, they call it strain B31, that the government and universities and commercial labs all have used from that day forward as their standard for Lyme testing. Now, a lot of problems with that. First of all, it came from a tick, never a human being, so it never tasted human blood. Second thing is, a tick strain that came from an island off of New York in no way reflects the cases of Lyme that occur in all different parts of the country, because we know that the Lyme spirochete has variable types and is often geographically located, and there are European strains, there are Northern American strains, there are Central American strains, East Coast strains, West Coast strains, Gulf Coast strains. So to base all the blood tests and all the research on this one thing that came from a tick from New York is a major, major mistake. And that's why, and I'll show you some evidence for this, that's why the testing that's based upon this until now has been so poor. The other thing is that they never considered co-infections, so that was another big vacuum in the testing in the beginning. Here, the second slide is the co-infection slide. In 2018, the Agenix lab collected 10,000 patients, actually more than 10,000, and they just tabulated the positives. And as you can see here, 37% were positive for Babesia, 32% for Lyme Borrelia, 27.5% or more for relapsing fever Borrelia, then a lot for Bartonella, Anaplasma rickettsia. And the point is that 40% had 2 pathogens, 15% had 3, 4.5, 4.6 had 4, and some even had 5 at the same time. And these are those who tested positive. And some of the tests don't always pick up, so it's even a higher percentage. So it's interesting to note that if you combine the two Borrelia, they turn out to be, as a Borrelia group, the number 1, and Babesia is number 2. But also, relapsing fever Borrelia, which is not at all reflected in that first tick sample that Willy Burgdorfer collected back in the 80s, that was never even considered. So I'm going to go through the basics of testing. And there are 2 types of tests available to the world of Lyme and tick-borne disease. They're indirect tests and direct tests. So starting with indirect testing, what is that? Those are the standard and the advanced serologies. The standard serology is what's called an IFA, the immunofluorescent antibody assay, the ELISA, enzyme-linked immunosorbent assay, and the Western blot. I call these standard serologies because these, by and large, are based on that lab strain that I talked to you about and don't reflect the real-world experience of real Lyme patients. The advanced serologies are those that Igenix has pioneered. The simplest one is called the Lyme Screen Assay, the LSA. That's their first tier in the 2-tier testing. The broad coverage assay is a much more advanced test than the ELISA. It replaces the ELISA because, as you can tell by the name, it doesn't just look for that lab strain, it looks for all the important strains. They have developed now the Acute, which is the broad coverage assay, which can be done as an at-home test kit. We'll talk about that. And then they're very famous for their Immunoblot, which has pretty much revolutionized Lyme testing. The other indirect test that we have is the T-cell response assay. And let me go through these one at a time. So an indirect test, what does that mean? It means they don't look for the germ itself. They look for how the host is reacting to the infection. I mean, the thinking is if you have an infection, your body's going to recognize it and react. So the tests are measured or are designed to measure that reaction. So there are 2 types. There's a humoral immunity. That means antibodies, liquid antibodies that are formed by the B cells. That's called humoral immunity. And the test measures the level of these antibodies. And that's what a serology does. It measures the liquid antibodies, the IgMs and the IgGs. The other part of the immune system involves not the B cells, but the T cells. That's called cellular immunity. And T cells are regulatory, and they also are the Pac-Men who come and get the germs. So T cell testing can be done to look for activation of the T cells after they've been exposed. So B cells are the humoral immunity, the serologies, and the T cells are T cell response assays. Here, a positive test indicates that you've been exposed to the infection. Doesn't necessarily mean you have the infection now today, but it does show prior exposure. So even though it doesn't show active infection, the thing is that during the course of Lyme and the co-infections, the B cell and the T cell responses do vary. So you can actually get a good feeling for what's going on with treatment. For example, if treatment is working, antibody levels generally rise, then they level off as the treatment is accomplishing its goal. And finally, as you're starting to recover, the levels diminish. And over time, if someone is fully recovered, you expect a negative serology. Now, this may take a year or more, but if someone a year out is still got a positive serology, I as a clinician would be very concerned that something is still going on. And this is another important slide. These different parts of the immune system react differently based on the time of exposure. Interestingly, the T cells after tick bite are the first things to respond. A T cell assay may start to show positive in less than a week. So if there's a very early case of Lyme, you just got a tick bite, you want to know testing immediately, what do you want to do? The best test for that is a T cell response assay. The IgM response is the B cell response of serology. That response next can start to show up a couple of weeks into it. It will peak early on and then diminish. In most people and with most infections, the IgM response goes away and it turns over to the IgG response, which is the third bump you see there. But as a lot of you know, and I'll talk about it later, in a lot of Lyme patients, the IgM doesn't go away. It stays a persistently positive. It had been thought that a positive persisting IgM, especially if IgG is not present, has to be a false reading because it's impossible to think an IgM would persist. But we found that in the Lyme patient, it is a true thing. You can have persisting IgM. That occurs because the infection kind of does things to your immune system. It prevents this flip from M to G. So IgG then comes next, and it usually persists throughout the course of infection, and only after it's controlled will the IgG go away. In some people, the T cell response, although it's early and goes away, will start to come back, especially in chronic Lyme disease. And it's an interesting relationship there because when the B cells, when the serologies are weak, the T cells are often strong. So it's like a reciprocal relationship. And that's why it's often useful to do a T cell test, not only in the very beginning, but toward the end of the infection as well. What about antibiotic treatment? Will antibiotics affect this type of a blood test? The answer is basically not. If you have antibiotics or you were given antibiotics and you are being tested, you can do a serology, you can do an immunoblot. It's not going to really affect it. There's some exceptions to this, and it's very important. First exception, they found that if you get antibiotic treatment very, very early, let's say you got the tick bite, you got a rash, start to get fever, you start an antibiotic right away, you may prevent the development of a positive test because you intervened very early. However, the rub is that even if you did not get enough treatment to kill the infection and you're still infected, if it was a treatment failure, you're still not going to get a positive test. Now, think what I said before, the antibody test measures your immunity to the germ. So in this case, it's possible that you never really developed a good immune response. And in my practice, I saw a lot of people who had late Lyme who were chronically sick and never responded well to treatment. And very large percentage of those, in fact, the majority were people who got very early treatment that was inadequate, like low-dose doxycycline, for example, or 10 days of something, or a single dose of doxy. They never got a really good, strong antibody response, and their infection was really hard to control. The other exception to this is how, again, the infection changes the immune response. In the very early part of the infection, you may have a false negative, or you can have a low positive antibody response and a very low serology. But if you go on antibiotics, what happens is it frees up the antibodies and you start to now show a positive or a stronger positive if it was weak to begin with. And in fact, I tabulated this and I have a seroconversion rate of patients who had a negative test in the beginning, started treatment, and as they started to improve, it didn't have to be at the end of treatment, but after a few weeks, few months, 36% of the time, the conversion went from negative to positive. So, as I said before, antibiotics generally don't really affect the test to the point where you say, gee, I can't take the test because of my antibiotics. No, you can take the test, but keeping this in mind, it'll help you get a more accurate result. Now this, what I just explained, goes to, refers to the B cell response to serologies and not the T cell based assays. Now, serologies are always the first choice because they're readily available, they're relatively inexpensive, you get results pretty quickly. And the shortcomings that I'll describe pretty much have been overcome nowadays, or at least most of them, because of the recombinant technology that IGeneX has pioneered in this space. Now, I also have other things that I mentioned, extensive research. I was very impressed by how, unlike some of the big labs that use a test kit from Germany or from some commercial place that makes the test, gets FDA approval, and it's never refined. I know that IGeneX are constantly redoing their test work. They're constantly retesting the samples, constantly improving the things. So that's really important. Another thing that has helped is that there's a recognition now that it's not just that one tick germ from Long Island, New York, on an island. It's a lot of different organisms, a whole diversity of organisms, and designing testing now that will accommodate that and look for all these other organisms have made the test much more accurate. And the ongoing validation improvements are based also on the fact that a lab like IGeneX, they use not tick samples and not commercial samples you buy from some lab supplier online. They actually use patient samples to develop their test. So that's why it's a real world accurate test. Now, I mentioned that IGeneX made famous the recombinant testing, which is what they use in the immunoblots and other assays. What actually does that mean? If you look at how tests are normally made when they don't use recombinant technology, they take the Lyme spirochete, for example, in Lyme disease, and they break it up into a lot of little pieces, and they use those pieces to make the test. But the problem is that when they break up these germs, they get a lot of non-specific chemicals, as well as things that will cross-react with other germs. So you get tests that are not very sensitive and not very specific. So what IGeneX has done for recombinant antigens is that they reverse engineer it. They look for the genetic code that makes these different proteins. They take that and they clone it into a bacteria like E. coli using a virus vector. That then E. coli produces purified proteins that they then purify further with a lot of different lab techniques. And they end up developing these immunoblots and broad coverage assays and AccuDarts, which allows them to detect multiple species and multiple pathogens with much more sensitivity and specificity. And we'll go through the data in that too. All right, one at a time, we'll go through the standard type of testing. We'll start the standard test called the IFA, immunofluorescent antibody assay. It's standard old technology, not recombinant. What they do, they take the germ or parts of the germ, they smear it out on a slide, then they put patient serum, and using a fluorescent marker dye, if the patient's blood has antibodies to the germ that they put on the slide, it'll stick to the slide and fluoresce under a black light. So that's really very crude. It's read manually, not very exacting. And I remember years ago when these tests were developed, they even said that the quality and age of the bulb in the microscope can affect how accurate the reading is. Because of all this, it's known for having a lot of false positives and false negatives. Sensitivity is at the very best 70%, but more likely 50%, but you can get at least a 25% or more false positive. Okay, so none of they're false negatives, but you can get what looks like a positive test when in fact isn't. Another big drawback, because they make it from the germ, it's one species per test. It doesn't account for the broad types of germs that are out there. The ELISA test has kind of replaced the IFA, and basically it is an IFA, but it's done by a machine, so you don't have to sit with a microscope. But they do the same basic technology, it's just read by a machine. Again, it's a single species, it's not a big advance in technology. Also, you might find some labs now are promoting they have a microarray or multi-well technology. Basically, it's an advancement over the ELISA, but still an ELISA. It's still a single species, and it's really not any more sensitive. In fact, publications show that as pretty much the same as the standard CDC ELISA, which is basically 50%. And the specificity is also not very good, so you get 20 to 30% false positives as well. And here's some data I want to show you. This is a compilation by Dr. Stricker published in 2007. Number of different labs here, 9 of them, got together and they actually tested the accuracy of the ELISAs. And they found that the sensitivity averaged only 49%. Basically, it's a coin toss. It's 50-50 whether it's going to pick up the result. Now, if you look here, I got an arrow to show. I highlighted in yellow. Here, this one test had a higher sensitivity, but the specificity got poor because there's always a trade-off. If you make the test more sensitive, you're going to get false readings as well. And that's one of the disadvantages, one of the other disadvantages of this type of old technology. Now, the Western blot is another one that people have used thinking it's a bit more accurate than the ELISA, and it might be. But again, it's not recombinant technology. What they do is they take a Lyme germ, in this case Lyme disease, they'll break it up using sound waves, they sonicate it to release all the different protein antigens that get released. And then they put it on a piece of paper and they subject it in a water bath to electric current. And the different proteins that are released and other antigens that are released from breaking up the spirochete, they migrate along the piece of paper based on their electrical charge and their molecular weight. In other words, bigger molecules don't move as much as the smaller ones. So when they print this out, you get this picture. And these little dark dots are where these antigens ended up after this electric charge, after electrophoresis. And these are what are called the bands on a Western blot. But, you know, number one, it's based on a lab strain. Number two, it's one species. And number three, where this ends up is dependent on the electric charge, and it's not very accurate. Here on the far side is a control, and here the other ones, not all of them exactly line up with where you'd expect them to line up. How dark the spots are based on how good the culture is, what they grew the initial germ, and the strong one gives you a darker spot, a weaker culture or a poor culture gives you a more fainted spot. So it's hard to tell a good result from this. And finally, how do you know what's a positive? A dark spot is certainly a positive, but how about the faint ones? How do you call positive and negative on this? It's very, very much guesswork. Again, the sensitivity, like an ELISA, is 50 to 70%. Specificity, because you can actually look at what the results are yourself, can be a little bit higher because it's interpreted by the practitioner, but still not so good, still a single species. So to get around all of this, IGeneX started to use recombinant antigens. And they came up with the first test called the LSA, the Lyme screen assay. If you're doing two-tier testing, this is what's tier one. And they use it in conjunction with the immunoblot for tier two. Recombinant technology, it totally replaces the IFA. The sensitivity here on the slide shows 89%, but I just looked it up. Actually, for disseminated and late Lyme, the sensitivity was 100%. It was like 75% for early Lyme. So the average is 89, but aside from early Lyme, the actual validation study picked all of them up. Specificity here shows 94%. Another bit of data I saw showed a 96.5% specificity. So in either case, it's far, far better than IFA, ELISA, Western blot, and so forth. They now have what's called a broad coverage assay. It's a simple replacement for the ELISA. And you can use this as a standalone test. You don't have to do an immunoblot with it. And again, it's multi-species, it's recombinant, and you can get all the different species of Lyme and relapsing fever Borrelia that I mentioned in the beginning. Unfortunately, it's a very simple test. It's either yes or no. It doesn't say what species they found, and it doesn't say if it's IgM or IgG or both. It's just a quick, inexpensive, relatively inexpensive way of getting a test result. Sensitivity and specificity are far better than the ELISA, and it's good for all stages of disease, especially disseminated and late infections. The immunoblot, that's the most inclusive and the most accurate serological test of any of them anywhere. It uses recombinant antigens, but not one or two, but many, many, many. I'll show you some pictures of their actual immunoblot. This does give you separate reports on IgM and IgG. It can detect multiple species and multiple strains and will identify most of them and give you the result. Sometimes you get an immunoblot and it says Borrelia species were detected, but it doesn't say which one, or Bartonella species doesn't say which one. Just because they can't identify the species, it doesn't mean it's a negative test. It's still positive. It's just that there's a species present that they couldn't identify, either because it's an unusual one or because there's more than one present. If you have a co-infection with several Borrelia at the same time, or several Bartonella or whatever, that can give you an unspeciated result. So if you get that on your report, it doesn't mean it's a negative. It really is a positive. It just doesn't say which germ was there. Now, This is a game-changing test because it's the only test found to be sensitive enough to use for early Lyme disease. They found the assay picked up 93% of early cases, and this is based on samples given to them by CDC. Now, there's never ever been a test in the history of Lyme that you can use for early Lyme disease. How many times did someone come to a walk-in clinic or a physician's office, was just exposed to ticks and they're starting to feel poorly, but there's no known bite, no known rash, but you say, well, is it a summer flu? Could it be a tick-borne disease? Should I give antibiotics? Should I wait and see what happens? Maybe the patient gets sick and then they'll come back. Or if there's a rash, but it's not really a Lyme rash, maybe, is it a spider bite? You're not really sure. Now, instead of watching and waiting or using an antibiotic, maybe that wasn't necessary. Here you have a 93% pickup rate for early Lyme, so it's completely game-changing. Here, of course, you can use it for all stages, early, like I described, disseminated, and chronic Lyme as well. Now, typically, the validation results based on the different tick-borne diseases, the sensitivity at worst was 90%. In some cases, depending on the series, it's 100%. In other words, it picked up all the test cases. And the specificity in terms of false positives was 1 to 3% at the most. And so this is available for Lyme, Borrelia, for relapsing fever, Borrelia, Babesia, and Bartonella. Just a graphic to show you. You saw the picture over here of the Western blot. It's based on a culture of a Lyme germ, in the case of Lyme disease, that's broken up and then spread out on the paper. And it's very messy, very hard to interpret, in my opinion. This is the immunoblot. When they make these recombinant proteins, it's laboratory created. And what they do, they don't smear it out on paper with electricity. They use an inkjet printer type to print it in exact spots and exact amounts. So you see how much cleaner that is, how much easier that is to interpret. So it's a graphic example of how much better it is. Now, the T-cell test is the other indirect test I wanted to mention. It measures not the liquid or humoral response, the B-cell response. It measures T-cells. A positive result indicates exposure. It doesn't mean you have it now, but exposure. It develops faster than the B-cell response, as I mentioned, and it does respond separately. So if you're seronegative, the B-cells don't react, but the T-cells do. You can still get a positive on this, and you would believe it. It's recommended for detecting very, very early infection, like I showed you in that little graph, as well as for chronic disease and in patients who are seronegative. The other trick to this, as you can understand from that first slide on the immune response, the two parts of the immunity, the B cells and the T cells. We know that people with tick-borne diseases often have weakened immunity, and sometimes it makes it hard to get over the illness. So it's important that you can use combination of a B-cell test like an immunoblot and a T-cell test like this one together to see how well the patient's immune system is responding. So in other words, not only gives you results of exposure to an infection, but how well your body is responding to that. And you can get this test for Lyme, Borrelia, and Bartonella as well. Now, we'll talk about the direct tests. There's a blood smear that you can do for acute Babesia only, not chronic, but acute Babesia. There's a FISH test. You can use that for Babesia and Bartonella. PCR is a DNA detection test, polymerase chain reaction. There's the culture now that you can get from IGeneX, and also the urine antigen assay, which was developed back in, back in the early 90s by the National Institute of Health, and now available a version of that through IGeneX. So a direct test, it actually is designed to test or detect the organism itself or parts of the organism in the person. So a positive result means that the person was infected at the time the test was taken. The exception to that may be a PCR because in the blood, PCRs indicate presence of the germ's DNA. In the blood, the DNA, foreign DNA, doesn't last too long. The body gets rid of it over a matter of days. The exception to that is if you have an infection in the tissues. And that's why you can do PCR testing of murder suspects or murder victims, of mummies even, of fossils, because PCR in tissues does stick around. Now, because of this, a lot of people who are Lyme denialists say, well, a positive PCR doesn't mean you're sick now, especially if you do a PCR post-treatment. I think you can't possibly still be sick. No, but a PCR in the blood, if it's positive, means you're infected. In the tissue, maybe not. So that's the exception, and it's important you know that. So people who do PCRs and deny the result, this is probably why they're trying to pull the wool over your eyes. Now here, unlike serologies, antibiotic therapy, be it herbal or antibiotics, will decrease the amount of germs in the bloodstream and can decrease the test sensitivity. So if you're going to use a direct test, it's best to do the direct test before treatment or during a big symptomatic flare where the germs might be present in high enough amounts to be picked up, or especially during treatment pauses if you're going to get a medication break and see how you're doing and you want to get retested. The only exception to this with a direct test is the Lyme urine antigen test that I mentioned before, because in this case, with symptom flares and with antibiotic therapy, more germs get flushed out and the urine test will detect them in a higher amount. Now, when is this test most sensitive? Very early in infection, before the full immunity develops, before the body can fight the germs off they're present in larger amounts. It's very good in people who are immunocompromised because there are more germs present. The immune system's not fighting them off. And again, in more severe infections, for the same reason, there are more germs present in the blood. Starting with the blood smear, that's only done in hospitals for acute babesiosis, none of the other ones. And it's not very sensitive. They rate the sensitivity by limit of detection. And here that means that here the limit of detection is 0.5%. So it means that you can find these only when 0.5% or more of the red cells are infected. They count red cells, see how many of them are infected. And if it's 1 out of 100, that's 1%. It's 1 out of 200, that's 0.5%. And that's as good as it gets. Turns out with babesiosis, the germs quickly diminish in number. So it's only useful to do a direct smear during the first week of infection. After that, you really get no value out of this test. The other thing is it can detect dead organisms, because even though there's an immune system, Babesia germs, even after they're dead, they still circulate for a while. So if you're relying on a Babesia smear, and it may still be positive after the Babesia are dead after treatment, and you may be getting more treatment than you need. But again, it's a blood smear. It's labor intensive, it's time consuming, and the accuracy is hard because it depends on the person doing it, the microscope, how much time they spend, and so forth. A much better blood test that's a direct test is the FISH. It's called a fluorescent in situ hybridization assay. Basically, it's the same type of a test as a blood smear. They put a drop of blood on the slide, but instead of using a standard ink-based stain, they use a fluorescent dye which is tied to an RNA probe. So it looks for RNA, the nucleic acid of the germ itself, in this case Babesia bartonella, and it will attach to that. So number one, it's very specific. You're not going to get a false reading from some other thing in the blood. In fact, the specificity has been found to be 100%. And here the limit of detection is not 0.5%, it's 0.001%, 100 to 1,000 times more sensitive than a standard blood smear. Now, the very important thing is it can detect organisms even if they're hidden and embedded in a biofilm. That's very important for Babesia and Bartonella because those do hide in biofilms. The other thing is, unlike a DNA test, which is a PCR, the FISH test looks for RNA, and RNA does not persist after death of an organism. In fact, they hang around for minutes to hours and then they're gone. So here, a positive result is a strong indicator that there was an active infection at the time of the blood test. In other words, they're not going to find this in a mummy, okay? If you have a positive FISH test, you were infected at the time the test was taken. What IGeneX does is the RNA probe that they use is a genus level detection. So it's not just Borrelia burgdorferi, it's all Borrelia. It's not just Bartonella Hensley, it's all Bartonella. And again, it's for Babesia and Bartonella. It's not for Borrelia, but they do use that experimentally. Just some pictures. And this is, again, a genus level test. Here, if there's a probe specific for one type of an organism, you're going to miss others. Here's a probe specific for MO1. And it did not light up for Microtia duncani. Here's one specific for duncani. It lit up, but did not for the other ones. Here's one specific for Microtia. It lifted or showed up for Microtia. Here, what they did is a genus probe where it's all Babesia. So you see this one probe that they use picked up all the different Babesia in the test. Pretty cool. Here for Bartonella, same type of thing. Here's Bartonella Hensley, here's Quintana, and this is a negative control. And all the positives were definitely confirmed by genetic sequencing, so you know that this is a real result and not just a false positive. All right, the other direct test is the PCR. That again detects the nucleic acid, the DNA in a specimen. It's very low sensitivity in the blood samples for a lot of different reasons. Number one, the germs in the bloodstream are there in very low numbers and may not be enough to be picked up by this type of test. And the other reason is that there are chemicals in our bloodstream that inhibit this PCR test reaction. They call them PCR inhibitors. And you can have a false negative result because of that. On the other hand, a positive PCR is very highly specific. So it's not a false positive. You believe a positive, you don't believe a negative, in other words. So what IGeneX has done to improve the sensitivity of the PCR is that they test both the whole blood and the serum, because some of the organisms like to live in the cell faction by the platelets and by the red and white cells. Others like to live in the free circulation in the serum. And knowing that, what they do at IGeneX is they test both of them separately. If they're testing a PCR for Lyme Borrelia, what they do is they look for not just the basic DNA of their chromosomes, but also the DNA of their plasmids. So in other words, if you get a Lyme PCR from IGeneX, you're actually getting 4 separate PCRs. They also do basically the serum and the whole blood for the other organisms that they do PCR with. Here again, it's a genus level detection, so you get all the different species. And besides blood, you can test other body fluids and tissues, in the biopsy as well. And I mentioned before the controversy whether positive PCR reflects active disease. If it's from the blood and it was positive, yes, it's active. If it's from a tissue, a biopsy, or a skeleton, obviously it's not. You can get PCR for Lyme and relapsing fever Borrelia, for Babesia, Bartonella, Anaplasma, Ehrlichia, and the Rickettsias. The revolution that came from PCR work has been now the culture. IGeneX is the only lab to commercially offer a culture for the tick-borne diseases. And what they do is they take a blood sample, they put it in an incubator for 2 weeks. In the process, they remove the inhibitors that are in the bloodstream and they allow the organisms to grow. When the 2-week culture is done, they test the culture by a specially advanced PCR that they made that they found in testing has been shown to be 100% specific. So here a positive result is very, very highly accurate. Again, it's a genus level test so that you're going to get complete coverage, including finding rare and atypical organisms I can tell you about a little bit later on that they found some really strange things in people. Here, the sensitivity is at least 10 times better than that of a plain PCR. And in fact, I've seen some data where some of the results were 100 times more sensitive than a PCR. And here it's available for all the major tick-borne diseases except Rocky Mountain spotted fever. The urine test that I mentioned before, here you see the picture from the NIH, that this is a patient's urine. I think it was, if it wasn't one of my patients, one of the patients from the East Coast. And it actually shows a whole Lyme spirochete in the urine of a patient based on electron microscope. And now they find whole spirochetes, they found fragments of spirochetes. So basically, when there's an active infection of Lyme disease, it's possible you can get Lyme germs or pieces of Lyme germs present in the urine. So that's the basis of the IGeneX urine test. They call it a Lyme dot blot assay. And it's available for Lyme, only Lyme though, but it is all the different Lyme Borrelia. So it turns out that when you're actively infected, you spill this into the urine. Not every day though, sometimes it's there, sometimes it's not. It's there more likely when there's a symptom flare, for example, during a Herxheimer reaction, during a lady's menstrual cycle. And so what we used to do on this very often is provoke a Herxheimer reaction by giving antibiotics for the first 5 days, days 1 through 5, and having urine collected on 3 different days, on days 2, 4, and 6. So if it's for initial diagnosis before someone's on treatment, that's the way you do it. If someone's already on treatment and you want to test during a symptom flare. It's very simple. If there's urinary tract infection, though, that gives it a false reading, so you want to make sure there's a urine culture done at the same time. So if you get a positive urine test for Lyme, you know it's not a false positive. 2 new tests from IGeneX. The Acute, it's an at-home tick-borne disease detection test. You can get it for Lyme, relapsing fever Borrelia, Babesia, and Bartonella. And they now have the iDart, which is the FDA-cleared immunoblot kit. It's an IgG test kit. We'll go through these. Now, the AccuDart is really big news because it's based on the IGeneX broad coverage assay. It's multi-species. It's recombinant technology. And it's very simple. It gives you a yes or a no answer. It doesn't name the species. It will say Borrelia positive or Bartonella positive. Doesn't report IgM or IgG, but you get a positive or a negative 1, 2, 3. They found the sensitivity was 90%. And the only test has a higher sensitivity than the AccuDart is the immunoblot. It's available for Lyme disease, tick-borne relapsing fever, Babesia, and Bartonella. And you can order each test individually, or they have a kit, a test panel, they call it, where all these are included in the one kit. So that might be important to do, because by getting the kit, there's separate 19 separate markers are assayed during that. And this is the thing. You buy the test kit online, acute.health.com, you do not need a doctor's order. Now that is really cool, I think, because, I mean, you know, if you're waiting for an appointment and you are a new patient and you have to get a testing done, you can get the test before the appointment. If you're getting a follow-up, you don't want to have to go back to the office. You want to do something at home. It's much easier. If you're ill and you're worried about your family members, get them tested. You don't have to get a doctor's visit or a doctor's order for this. So, I mean, that's revolutionary. And all it is a simple finger stick. Get the kit from Acute.health. You get the finger stick done. You use a little lancet and you mail it off to hygienics where it gets processed. You know, doctors can also keep these test kits on hand in their office. So if they don't have a lab nearby, they can at least get the blood done this way and not have to worry about needles and blood draws and the whole thing. And if you know you want more information than just a positive or negative, you can always go and order the Immunoblot and get a full panel on it. But it's really a revolutionary test that's very highly accurate, very inexpensive compared to what else is out there, and so convenient. Now, the Immunoblot being cleared by FDA is also very big news. What does that mean? Laboratories can develop their own tests, which is what the Immunoblot has always been, developed by hygienics, but it means that it can only be done by hygienics. If you turn it into a test kit that gets FDA cleared, that means that hygienics or whatever company, and this company is called IDART, they can sell this to other labs or other hospitals or other institutions. So now that means your local hospital, if they want, they can order this from the company and in their own lab do the Immunoblots. So, because it's FDA cleared. So number one, that means a high quality test like this is available much more broadly. In other words, before, hospitals would have to get one of those CDC type of FDA test kits that is so inaccurate, 50-50 pickup of one species. Here they can get a full Immunoblot if they want to do it. So I recommend you go to your local hospitals and get them to order this kit. The other thing that's really cool, it's a 2-tier test. So this already will cover you know, what the CDC wants. It also will cover what a lot of insurance companies require if they want to cover your treatment. They want to have a 2-tier test. So it includes the Lyme screen assay and the Immunoblot in this one kit. So that's really pretty cool. Now, because it's FDA cleared, does that mean it's covered by insurance? Well, it's not up to the lab. That's up to the hospital or the institution that actually does it. If you go to your hospital and they accept your insurance for their testing, if they order the Immunoblot from IDART and they do it at their own institution, then it's up to them whether or not they want to take your insurance. But I'm pretty optimistic. Now, another very important thing. Those of you who know about Western blots know that in their crooked wisdom, the powers that be in government did not allow bands 31 and 34 on the Western blot for political reasons mainly, and that made the test so much less accurate. Here, however, the FDA has allowed hygienics to include bands 31 and 34 on the Immunoblots, and the FDA has now allowed that on the test kit IgG Immunoblot. So it's the only FDA test kit that includes these two bands, which are critical bands in Lyme disease. The other thing is that this is interpreted using hygienics criteria, not the CDC criteria from the Dearborn Conference. What does that mean? You don't have to have 5 bands. All you need is 2 separate bands. If you get a positive LSA screening test and you do the second part, the Immunoblot, all you need are 2 bands from the different groups, and I'll show you what that means. This is an actual picture of the IDART Immunoblot. What this has is groups of tests. So on a Western blot, you might have 1 band for 41, okay? You might have 1 band for 23. Here, they have 9 different 23s on the Immunoblot. So this is why it can cover so many different species. They have 9 different bands 31. Western blots don't even have 31. Here they have 9 of them. They have 2 39s, and a lot of you know 39 is the most sensitive test for Lyme. They have 2 41s, and 41 is a flagella band, but it's recombinant. So these 41s are specific to Borrelia, not cross-reactive. They have not 1, but 2 different controls. So in addition to the controls, a total of 31 different bands on this. So to get a positive, all you need is a positive from one group plus another group in addition to this Lyme screen, which is this basic tier 1 screening test. So, I mean, this is completely revolutionary, and hopefully a lot of hospitals and a lot of commercial labs across the country will start offering this test kit because it's so much better than everything else that's otherwise out there. And it's kind of revolutionary that they got this covered. All right, let me finish up by talking about when do you do which test, okay? So here, these are the Lyme, Borrelia burgdorferi, Lyme germs that are known to cause Lyme disease. The one that was found by Willy Burgdorfer on the island off of New York was called B31, and they call that Borrelia sensu stricto, strictly Borrelia. But we now know there's other Borrelia burgdorferi, there's Californiensis, Mayonii, Afzelia green, many different other Borrelias. This whole group together, they call it sensu lato. So if you ever see BBSS, it's just this one thing. If you see BBSL, it's the whole entire group. In fact, there's a proposal to call this whole group Borreliella, not Borrelia, just to distinguish it from relapsing fever. When it comes to relapsing fever, there are tests you can get in commercial labs for this one called Borrelia hermsii, which is not that common. Miyamoto is another one that's been discovered that you might have heard of. But all these other relapsing fever Borrelia can affect Lyme patients and actually mimic Lyme. So the key here is that those in red are the only ones you can get from a commercial lab. And in the case of relapsing fever, you have to order these two tests separately, and they're both very low quality. This is an IFA, and this is a single band ELISA. It's useless. But IGeneX includes all of these and all of their tests. So that's why it's so much more logical to use IGeneX than the commercial lab. All right, so for Borrelia early disease, an atypical EM, a summer flu, the Lyme and relapsing immune blot have a 90 to 93% sensitivity in early Lyme. The T-cell response assay is also available for Lyme. It's very good early on. And the culture is also very good because spirochete load is very high early in the disease before the immune system matures. And that's for Lyme and relapsing fever Borrelia. With disseminated Lyme, let's say you have it for a few months, not to the chronic horrible state with an intact immunity, immune blot is always the first choice. You can add the culture to it to increase the yield. Newborns and patients who have trouble getting blood drawn, you can always do the IGeneX urine test. When I had patients who had Lyme during pregnancy, when the babies were born, I would have a urine antigen test done on the baby at birth and every month for the first 6 to 12 months, because nobody, especially babies, like to get their finger stuck. For chronic Lyme, especially those with co-infections and immunosuppression, you always do an immunoblot and you add the culture because that's an indirect test, the best indirect test, and the best direct test. And they did studies and they found that when you combine the two, you get a much higher yield. You can also add the T-cell test, as I mentioned, to give you a better idea of the whole immune response. The B-cell response is an immunoblot, the T-cell response is a separate test. And so that way you get not only results of your Lyme, but you get results of your immune system. And again, it's always a good idea, if you need to add the urine test, the dot blot assay, especially during a symptom flare. For Babesia, they're very hard to detect, as you probably know, because the lab testing is not that great. The reason why is that circulating Babesia, they rapidly decrease. They get sequestered in organs, in biofilms, stuck in blood vessels, and the direct testing is not always that great. Because Babesia can be immunosuppressive, it can impair the indirect tests as well. And finally, there are many, many different Babesia species, and you want to have broadest coverage. So the solution here is to combine methods, again, to achieve the highest yield. For the early infection, if you're going to do a blood smear, it's only for the first week. A fish is far better, as I showed you, 100 to 1,000 times more sensitive. Immunoblot reactivity can appear within the first 2 weeks. So early infection Babesia within the first 2 weeks, that's another option. And the culture is also a very good choice because not only is it a high yield, but you get a broad range of species detected. I'll show you some information on that. If I have to go back to this, there was a patient who had a diagnosis of chronic fatigue syndrome for several years, different testing of many different kinds, could not pick out what it was. So she ended up getting a Babesia culture, and it showed an organism called Culpadella. Culpadella is a one-celled parasite that lives in soil and in ponds. And somehow it got into her and made her chronically sick. The lab didn't believe it, because who ever heard of this? So they sent it out for sequencing, and it was positive for Culpadella. So they didn't believe it again. They'd sent another test. Again, it was positive. Turns out They believed it. The clinician gave her treatment for malaria because it is in that family, and she got better. After years of being chronically sick, labeled as chronic fatigue syndrome, she got treatment, she got over it. So that shows you the power of the culture. It picks up not just Babesia, Bartonella, it picks up anything that might grow in that medium. In fact, I looked up the medical literature. There's only 2 cases reported in the entire world of this infection in a human being. And now I guess there's a 3rd one. All right. In disseminated Babesia and chronic Babesia, the most sensitive thing is to combine all the tests you can, because you really need to get that if you're going to get a pickup. Immunoblot for sure, adding a fish and or the culture is very, very good. Sometimes they can be missed though. So there are tricks. How do you increase the yield? This is some things I've learned. If someone is co-infected, they have Lyme and Babesia, because Lyme impairs the immunity, if you start to treat the Lyme first and then you redo the Babesia immunoblot, because treating the Lyme helped your immune system, now you can get a positive Babesia. On the other hand, the same thing goes in reverse. If someone is clinically presenting with Babesia, you can get Babesia treatment, and then as the immune system heals, a seronegative case of Babesia becomes seropositive. Another trick that a lot of clinicians are doing is they're adding fibrinolytics, Boluc and serrapeptase, to break up the biofilms, release the Babesia to make the direct test more sensitive. For Bartonella, again, it's the same type of stealth pathogen. They're very hard to detect. Notorious case of unknown fever, unknown culture negative endocarditis. There are at least 45 different species. So you want to have broad culture testing, broad coverage testing, and that includes all the things we talked about. Now, there's an article that showed Bartonella PCR by itself only picked up 6% of cases, a false negative 94%. So what's always recommended here is always you start the immunoblot, because it's broad species coverage. It'll cover the 45. It's got good sensitivity. It shows if your body's responding to the infection from an immune point of view. You can always add the T-cell test to see more information on immunity. Adding to that the fish and culture are the direct tests that increase the yield, especially in chronic disease. And we're going to finish with the rickettsias. You don't often hear much about rickettsias, but you should. Anytime you read a series of testing done on ticks, where they screen ticks in the wild and they see what's in them, the number one always is a rickettsia of some kind. That includes Ehrlichia, Anaplasma, different rickettsias. So it's the most commonly found pathogen in the ticks. If it's an acute case and not treated, it can be fatal. I have actually seen that happen in my local hospital. It wasn't my patient, but someone else was misdiagnosed or not diagnosed, and he passed away. And they're also finding unusual species. Here again, because of the culture. They found Anaplasma platus. That's an anaplasma that's supposed to only infect dogs, but they found it in human cases. Rickettsia parkeri, it's been in the news now. It's normally found along the Gulf Coast, but guess what? It's all along the East Coast, as far as Connecticut and yes, Pennsylvania too. So for Ehrlichia, Anaplasma, and the basic ricketsias, you can do a serology. They don't have an immunoblot, but the PCR, the culture, the CE-PCR is available for that as well. Only exception is Rocky Mountain spotted fever. The government doesn't allow culturing of this. They want a biosafety 4 lab, which, Igenics is not. So that you only can do a standard PCR. But if it's present, it will show up on the culture if it's being cultured for the other ricketsias. But again, because this can be a severe, even fatal illness, the advice is always use every test you can if this is being suspected. Pregnancy, you're detecting infection in the mother and the baby. It's a double infection. All the major tick-borne diseases can cross the placenta, but sometimes the placenta blocks it. So in this case, you want to test the mother and the child. So you must maximize. You want to do an indirect test. The best is the immunoblot with direct testing like the culture in fish if it's that kind of an infection. The baby, like I said before, you can test the urine because you want to do it at birth, and also you want to do it serially during the baby's first 6 to 12 months of life, unless symptoms show up or unless the test is positive, then you know right away. And finally, what's not done and must be done and should be done is the placenta itself should be looked at by a pathologist. Normally they don't. It amazes me that placenta is such an important organ, they throw it out. But it should be looked at by the pathologist because they can actually look under the microscope and see abnormalities that can predict autism, believe it or not. They should do a PCR and culture on it too to see what's in there. Now, can you use testing to see whether treatment is working? we don't have a test for cure. You really can't measure treatment response. It's not like you can count germs, but there are tricks. So as I mentioned before, during treatment, the serologic response may actually increase in the beginning if treatment is working. Later, the response levels off if the patient's getting better. If someone has only had a positive IgM, if the IgG starts to appear, that's a sign of improving health and recovery. The treatment is working. And again, as I said before, after treatment ends, expect the antibody levels to diminish over time and eventually disappear. And a positive persisting is a cause for concern. Last 2 slides, little testing guide. I'll have a PDF sent to Eric so he can hand this out to you. So very simple. The IFAs, the ELISAs, Western blots are serologies. They use organisms, whole organisms that are broken apart, only one species at a time. They're not recommended anymore because of false positives and false negatives. The LSA, the Lyme screen assay, is a serology using the recombinant technology. It replaces the IFA as tier 1 in the 2-tier test, very highly accurate. The broad coverage assay goes one step further. That's, again, a recombinant serology. Multi-species is simple yes and no. IgM and IgG are both together, and it replaces the ELISA. You can use this in all stages, and again, it's highly accurate. Immunoblot is a very highly accurate recombinant serology. It does name the species. It separately reports IgM and IgG, and it's multi-species capable, which is really important. Again, all stages and maximal accuracy. And the T-cell response assay, they call that a mitogen stimulation assay. Limited time window in the very early part of the infection and very late when someone is chronic. When do you use it? Again, early and late. And the accuracy depends on the timing. Final slide, the direct tests. The PCR detects DNA of the organism, fluids and tissues. When to use it, tissues only, because fluid is only a 6% pickup rate, as bad as it is. Again, it's very insensitive, but very specific. So if you get a negative, well, you got a negative. Get a positive, you believe it. I always use the analogy, if you're going fishing and you get a fish, it's positive, there's fish in the water. If you don't get a fish, it doesn't mean there's no fish in the ocean, it means you just didn't pick one that day. The culture is where they incubate the blood for 2 weeks, then they confirm the identification by a special PCR, can be blood and tissues, all stages, but not if you're on treatment. Maximal accuracy and able to detect these weird organisms that are showing up. The fish test is a RNA-stained blood slide, the best test to use if biofilms are present. All stages, but not if you're on treatment, pretty good accuracy. And the urine antigen capture is direct antigen detection, but it's only for Lyme. It's good to use when you can't do blood draws or you want to avoid them. And it's especially good to take this test during symptom flares. And finally, that's it. So if we have time left over for Q&A, I'm going to turn this back to Eric. And we'll see what's going on. You're muted.
Eric,
Microphone.
Eric, we cannot hear you.
Doing Zoom for 4 years. You'd think I would know by now to unmute myself. Thank you for that. I did not mention that if you have any questions, go ahead and post them in the questions and answer section. And for those of you that are watching on Facebook live stream, if you have any questions, go ahead and post them and we'll capture them. I got a couple of questions that were texted to me from some folks at the Harrisburg Lyme group. And the couple of them revolved around the same Dr. Burrascano, that if tests like Immunoblot are better in terms of sensitivity and specificity, why is it that it's still ELISA and Western blot that is promoted widely? When will a sea change happen where the testing will become better but more standard and normalized?
Well, you see, that's what's happened now with the FDA clearance of the Immunoblot. See, if you are a local hospital or if you're a big lab like Quest or Humana or something, you are obligated to buy a test kit from somebody because you don't have your own, incubator growing spirochetes and doing all this rigmarole. You want to buy a test kit, put it in a machine and press a button and get a result. And the only test kits that are FDA approved until now are the single species ELISA and IFA. Terrible, terrible quality testing, but that's the only ones that until now have been FDA cleared. So now the Immunoblot is FDA cleared. And now if the hospital or laboratory institution wants to get a better test, they can. I think that as word gets out, they're going to start to do it. And I know that, for example, in New York City, some of the bigger hospitals are now opening up Lyme disease clinics, and they're all in competition with each other. And they're going to try and compete for the better tests and the better treatment. So they'll be the first in line to buy these better test kits. And as I mentioned, you can go to your local hospital and say, look, here's the data. The testing you're offering is awful. You can now get this much better kit. Let's do it.
The free market will help to push that in that direction.
Yeah.
I had it explained to me that when somebody is bitten and they have a spirochete in the bloodstream, it's in the kind of the auger-shaped spirochetal L form. My understanding, as it was explained to me, is that when somebody takes antibiotics, that the spirochete will go into a cyst form and kind of play cat and mouse and hide. And as long as there is antibiotics in the bloodstream, it'll stay in that form. And then when the patient is off of antibiotics, the cyst form will then come back into the L form, but 10-fold, 20-fold. And it was explained to me, but I don't know if this is accurate. That's why I'm asking, that when the antibiotics is out of the bloodstream and it comes out of this cyst form and it goes into the L form 10, 20-fold, that those spirochetes have like a protein antigen that hide from the immune system. Is that an accurate representation that after antibiotics, when they come back out, they're going to fool the immune system because there's a protein shell on the outside, antigen to evade the immune system? Or is that a lot of not so?
Like everything in Lyme, it's a lot more complicated than that. First of all, the spirochete form is the one that has the most surface antigens, and it's what you get picked up by when you do antigen testing. It's also what the immune system recognizes and forms the antibody response. You get all the different bands on the Immunoblot because all the different surface proteins of the spirochete form. If someone goes on antibiotics early, or they've been on antibiotics for some other reason when they get the tick bite, right away, the spirochetes that are not killed will try and encyst themselves, go into a cyst. And when they're in the cyst, or even in an L form, they lose these surface antigens, or they're actually hidden within inside. So you're not going to get an antibody response, and you can get a false negative. Now, when they come out of this, they're going to have the antibodies again. So they can be detected, but that's going off antibiotics. Now, the question is, can they make a coating around themselves that can fool the immune system? Well, they do that anyway, whether you're on antibiotics or not. They call that an S layer, S for slime, believe it or not. And I can, in fact, that picture I showed you in the slide of the urine test for the Lyme spirochete, it was a fuzzy looking spirochete because it was coated in slime. So yeah, that's a real thing. And that's the basis of what makes the biofilm also. So what you described is kind of true, but not exactly in detail. But yeah, it does make a coating. And when it's in a cyst form, it becomes seronegative.
We do have one question from Facebook. Someone is asking, you mentioned the urine test is for Lyme only. Is that just IGeneX, or do other companies have a urine test for other pathogens?
Yeah, the other lab that does a urine test, both labs do multi-species of Borrelia burgdorferi. In other words, that list of Borrelia sense. I'm saying it the wrong way. IGeneX is the only lab that does urine testing that'll look for all the different Lyme Borrelia. Okay, the other one uses Stricto. But they both look for different bands. For example, in IGeneX, they'll do band 31, 34, 93. They have a whole bunch of different bands. In fact, all the urine tests do a lot of different bands, but IGeneX does multi-species, not just Stricto.
Okay, we had a question coming through Zoom. This person is saying they're part of a large COVID vaccine injury group, had tens of thousands of injured, testing positive for Lyme and co-infections, reactivated Epstein-Barr, as well as T or B cell clonality abnormalities. Do you know of these tick-borne tests creating false positives in the now immunocompromised vaccine injured, or should those positives be accepted as valid?
Oh, it all depends on which test, by what lab, and by what method. If it's a direct test, like a culture or a PCR or a fish, definitely it's positive. I mean, those don't usually get false positives. A serologic test, if it's not an immunoblot or a broad coverage assay, then yeah, there could be a lot of false positives. But if it's an immunoblot, the false positive rate is between 1% and 3%. So that's, very, very low. For an IFA, false positive rate may be 25 to 40%. So again, it depends on the testing. If you can get it from IGenics, I would believe a positive. And incidentally, as clinicians, since COVID, we've seen that. We see or activation of Bartonella and Lyme and other tick-borne diseases that we never knew the patient had, or maybe they had years ago, and it all came back out again. And actually, in my experience, the number one reactivation after the other DNA viruses is Bartonella.
All right, thank you for that. Someone is asking about the presentation, or is the presentation going to be made available to folks? Eric, I think that's for you.
We will be getting the presentation. We do not post it globally, but if you would like a copy of the presentation, please email me at ehuck@palyme.org, ehuck@palyme.org. This is being recorded. It takes us a couple of days to clean up the recording. We will post the recording on our YouTube channel. If you go to our website, palyme.org, and go to the YouTube channel, you'll see this recording posted there. If you want the printed slide deck, email me at ehuck@palyme.org.
If anyone else has a question, let me know. At the moment, I'm not seeing any more. I'll check Facebook once more. You could either use the Q&A feature in Zoom or leave me a comment in Facebook. Okay, someone has left another comment in Facebook. Prior to the immunoblot was typically recommended if one was on, let me read that again. Prior to the immunoblot was typically recommended if one was on treatment because it picks up bits of spirochetes, and shedding of them creates greater accuracy. Is this outdated or incorrect information?
No, it's true. I mean, I can get kind of technical. It turns out that when you have an infection, there are a lot of germs. In fact, there's more germs than antibodies. And you think of the antibodies and the germs as like Pac-Man. They try and eat each other up. And the germ will adhere to, or the antibody will adhere to the germ, and they come together into what's called an immune complex. Now, it turns out blood testing for infection, in this case, tick-borne disease, it measures the antibody, but only if it's free. If it's attached to the germ, you can't detect it. You get a false negative serology. So if there's a lot of germs, an overwhelming infection, all the antibodies are stuck on are bound into it, and you get a false negative. But as I explained, when you treat it and the number of germs decreases, now the antibodies are freed up, and you can start to get a positive. So yeah, treatment will break free some antibodies, and you can start to get, if it's a weak positive, a stronger positive. If it's a few bands on the immunoblot, you get more bands. If it's a negative, you can get a positive. So that's actually true.
All right, thank you. Any other questions out there, folks?
I see one, Bill, I don't know if you see that, the anonymous attendee, for the fish test. Where have you found a better level of accuracy between T labs and IGenics?
I think they're pretty equivalent. I've seen people have positive in one lab and negative in the other, and vice versa. So I can't say one lab is better than the other, but they are different. T-Lab does a fish test for Borrelia, which IGeneX does not do. They both do fish testing for Bartonella, and IGeneX does fish testing for Babesia, which I don't think T-Lab has yet. I think they're working on it. But again, it's not so much the labs, because both of those are very good labs. It's the situation. If the Babesia or Bartonella are stuck in a biofilm, it's going to be much harder to detect if the immune system is crowding them out, or if they're stuck in a blood vessel, they're sequestered. You're not going to be able to pick them up. So it really depends a lot on the situation, and that's why you can do the same blood test, once a month for 3 months and get 3 different results, because it has to do with how the germs change. So it's very hard to compare the 2 labs, but they're both high quality labs, and I would trust either one.
Okay, we got a few more questions coming in. Which AccuDart test do you recommend? The full panel test? How to decide which one?
Well, it's up to, see, that's a really good question, actually, because normally the physician or clinician or practitioner will say, gee, based on your symptoms, I think it's very rare that you would have Babesia, but probably have Lyme and Bartonella based on symptoms. But, the average patient may not be that experienced to know the difference. So they say, gee, should I do a Lyme test? And if I'm negative, then maybe I'll go do a Babesia test. If I'm negative on that, I'll do a Bartonella test. So to eliminate that kind of a guesswork, I kind of think it's better to do the whole panel, especially because at this point there's some kind of a discount on it. But on the other hand, if you know that you've had, let's say, babesiosis, and you want to follow up on that, you can get just that one test and not have to go for the whole panel. So it depends on your situation. What I can do for you guys is I have a table that lists the different symptoms of the different illnesses so that you can use a clinical guide to guide your testing. So I'll send it to you as a PDF also. You can include if people want it.
It's helpful, thank you.
Very good, thank you. If I have a friend who's wondering if they have Lyme and they go to their doctor who orders ELISA and Western blot, what are the chances that patient can ask their PCP to get FDA cleared test instead?
The chances are not good, and I'll tell you why. If you go, let's say I have in my neighborhood a LabCorp. So if I want to go to and get a blood test and I want to get a Lyme test and I go to LabCorp, I have to get a LabCorp Lyme test. They're not going to take my blood in their lab that they spend money on and send it to some other laboratory. So if your physician has a nurse or a practitioner in his or her office that'll draw the blood and pack it and send it, then you're fine. You can have them do that for you. You can call IGeneX and get the kit, or go online and get the blood test collection tubes and stuff, and they'll send it off. But most physicians, if they don't have someone in their office to draw blood, they'll send you to a local lab, and that local lab is not going to do that. That's where the AccuDart is so great, because you can do the test even before the doctor visit. Or you can say to them, I'm going to bring this in and we'll do the test in your office. Or the doctor can order the AccuDarts himself or herself and keep them in the office and just get them done that way. And that's why that's so revolutionary. And the technology is recombinant broad coverage assay, so it's really good.
I think this is just a follow-up to the same question. I know that you said hospitals and clinics will want to stock these, but what about PCPs or ID docs where most people go for testing?
Well, they can stock the iDart, but they'd have to have a dedicated lab to run an FDA test kit, like a hospital or so forth. A regular doctor's office doesn't have that.
Okay, here's another question. Would it make sense to take, oh boy, would it make sense to take a proteolytic enzyme like lumbrokinase, serrapeptase, or nattokinase before testing to reveal the pathogens hiding behind biofilms?
Yes.
Very good.
Easiest question of the day.
It's harder to get the question out than to get the answer.
I got a question here based on that one. Do you mind just taking a second and giving an overview of biofilms? What's your take on biofilm, and what should patients know about biofilms as it relates to testing and their illness?
Okay. All the major tick-borne diseases, that means Borrelia, Lyme, relapsing fever, Borrelia, Bartonella, and Babesia, all of them are biofilm producers. And what that means is that they can find a niche in an organ or along a blood vessel or both and settle in and start to secrete this gelatin-like substance around them. And that protects them from the body's immune attack and also makes them inaccessible to testing. So if they're coated with this gelatin, they're no longer picked up by the immune system. So antibody tests become less sensitive. If you want to do a direct test like a PCR, they're not circulating in the bloodstream. They're stuck on the wall or they're stuck in an organ somewhere. So that's why you'll use one of those enzymes that you had trouble reading, but you'll use one of those enzymes or a group of them to loosen up the biofilm and let some of the organisms escape. And then you can actually do a direct test, or in fact, any of the tests, but here a direct test like a FISH test, which does find organisms even if they're in a biofilm. So that's the trick there. There's no other way to really bust up a biofilm than using the enzymes. The problem is that not all enzymes reach the bloodstream. A lot of them get degraded in the intestinal tract. And, the ones that clinicians like the most include Boluc and serrapeptase, and they usually use them in combination. Now, the one caveat is that if you're very sick and a lot of germs are in your body sequestered in biofilm and you start taking these enzymes, you may release a lot of them and you may get a really bad Herxheimer reaction. So you have to be very careful. Don't do it on your own without talking to your clinician first.
Someone is asking your opinion on microscopy. This person is saying they've been trained in phase contrast Borrelia microscopy, and this person also knows dark field. And do you think we might see microscopy as a commonly used test by LLMDs in the near future? It takes time, of course, so manual labor is considerable. Just curious.
Microscopy is how Lyme was first figured out. But you can't just look at a blood smear, even with the dark field, and claim that we're finding it's definitely a spirochete, because there are things like cytoplasmic strands, even yeasts, protomyxoa, other things, biofilm strands that can mimic a spirochete. So at the very minimum, you have to use immunofluorescent dyes that, well, spirochetes specifically. You can go a step further and do a FISH test, which is where it binds to the RNA of the organism. But even so, direct smear is not always the best because you don't see them all and you can get false positive readings. And that's why they were replaced by FISH testing and by cultures. And back in the early days of Lyme, back in the 80s, a lot of people did dark field direct smears of the blood, and they made a lot of claims and diagnoses which were not found to be accurate.
All right, thank you for that. Someone's asking about the enzymes that we mentioned. Would either of you want to talk about, I'm sorry, how long do you, where can you get the enzymes is the question, and how long do you take them before getting the test?
Oh, you can get them online. I get my Boluc from Research Nutritionals, but you can get them from a lot of different places. And you generally want to be on them at least 5 days and maybe even as long as 2 weeks. It depends on how sick you've been and how your history of testing has been in the past. In other words, if you've had repeated testing that's negative, but you're clinically sick with that organism, then you might have to use a biofilm buster longer.
All right, here's another question from Facebook. Let me try to condense this a little bit. Should you ever trust IgG positive results as indicating active infection in a person who's been sick for 10 years and already gone through a lot of treatment? And why couldn't the symptoms or disabilities remaining? Could it be just something left behind from Lyme disease and the IgG will always be positive?
Well, let's try and figure this one out. In most every infection, once it's cleared, the IgG response will eventually go away, okay? However, IgG responses often persist in cases of, for example, getting a measles vaccine as a child. You might show positive serology for that for a long time. Hepatitis vaccine. So what's the difference? Well, in my experience with Lyme, you know, first of all, I don't know if you can actually 100% get rid of every last Borrelia. What you do with treatment is you get the number of germs below the threshold of how your immune system can handle it. And as long as you keep the balance tipped in favor of your body and away from the germs, then you're going to stay well, and you may still have a positive serology. If treatment is completely successful, the IgG, in my experience, will go away completely. Now, if you've had Lyme or tick-borne disease and you've been through treatment, but you're not completely over it, it doesn't always mean the Lyme is still there. There's so many other things that happen. Any chronic illness, and especially tick-borne diseases, can impair your body's ability for detoxification. And people who are chronically sick with Lyme almost universally are toxic from things like mold toxins, heavy metals, glyphosate, insecticides, other chemicals. And sometimes we see people who have been through Lyme treatment are not completely better, but then they go through detoxification and building up their membranes and all the complementary types of treatments, and then they do get better. So it's hard to attribute persisting symptoms to persisting infection unless you have a positive direct test like a culture or fish. And then if you're still symptomatic and you have negative testing, then it's time to find a physician who'll go that one step further and start to test you and treat you for the other things that happen when you get sick with tick-borne diseases.
Okay, here's another question on Zoom. I'm currently seeing a neurologist for CIDP. What information source can I guide them towards to accept the validity of these out-of-network labs?
Well, you know, IGeneX, for example, their validation data is online. You can see that they have a sensitivity of 97% and a specificity, whatever. I mean, it's there. And they have publications that are in peer-reviewed literature. So, I mean, it's out there. I would start at the IGeneX website. You can also go to the ILAD's website and you download the articles and the booklets and so forth.
Ready? I think we've run out of questions for now. If anybody has a final thought, still looking. I don't see anything.
One question came up. Should I do mycotoxin testing before tick-borne disease testing, or can they be done concurrently?
Well, that can be done concurrently.
I don't see anything further on Facebook.
It's a lot of information, a lot of detailed, specific information. We're, you know, very appreciative that you take the time to do this. And I'm getting some emails and texts from people that are looking forward to getting the slide deck. So thanks for sending that. And you said you have a grid as well that lays out some of the symptoms with what that'll be helpful.
Yep, I'll add that to it at the end.
That's good. All right. We appreciate you doing this for us very much so, and really just your leadership in this whole area with Lyme and tick-borne diseases. So thank you for being a part of our group again this evening. Based on the cycle, we'll see you again in 4 years. So
I look forward to seeing you sooner than that, hopefully. And thank you both for your leadership as well.
So with
Thank
that, thank you. So for everyone attending, we appreciate the audience, and we will talk to you again. Bill, thank you. Dr. Burrascano, thank you.
Have a good one, everyone.
Good night, everybody.
Bye.